hearing a term like the Black Death conjures up images of sickness and suffering. But for Hendrik Poinar, these pandemics present a mystery to be unlocked. So to tell us more about his research into these devastating plagues, we welcome back Hendrik Poinar. He's the director of the Ancient DNA Center at McMaster University and researcher at the Michael G. DeGroote Institute for Infectious Disease Research. Welcome back.
Thank you,
so last night we talked about woolly mammoths.
Yeah.
Tonight it's the plague, and we're ancient pathogens, and we'll talk about that. And we, I think we all studied various plagues, even in elementary school. Some have gone on before, but I think we need lots of refreshers. So
sure. Sounds
the
good.
Plague of Justinian, this means nothing to me in terms of recollection. So what was the Plague of Justinian?
So it was a major plague that basically wiped out the Holy Roman Empire, so about 1,500 years ago. So it spread somewhere from the Middle East, Egypt, we think, up through Europe, all the way up into parts of what's present-day Germany, France, even England. And it wiped out about 30 to 40% of the population at that time.
Okay.
So it was a huge plague, had a huge impact.
All right, and then comes the Black Death.
Then you fast forward 800 years, and then all of a sudden, out of what appears to be nowhere, something migrating from Crimea through Constantinople, through the Mediterranean, into Sicily, had the same thing, wrapped all the way back up into the Scandinavian countries, killed an approximated 40 to 50 million individuals, and that represented 40% of the population estimates at that time.
All right. Just because people say, what about the bubonic plague? That's the same thing.
That's the thing,
the
same
bubonic
thing.
plague, the plague, as
the
we call
plague.
it.
Well, the Black Death, for a long time, no one really knew what it was, right? It was called the Black Death actually much later. So in 1345, when it actually struck, no one actually referred to it as the Black Death. It was just because it led to social collapse afterwards, right? The people that were working the fields were dead. The landowners didn't have anybody to work the field. So it was actually a rebalancing of everything. Some people went straight to church because they hoped they would survive. Other ones lost complete faith because they lost their entire families. So it was a very dark period. And so the Black Death became this sort of
Okay, for purposes of our conversation, let's call it Black Death and the Plague of Justinian. So we got the two. They both ravage Europe.
That's right.
Kind of travel the same path.
That's right.
They both, at the times that they struck, wipe out about 40% of the population.
30 to 40%, yeah.
What do we know about, is there a link between them? Are they the same pathogens?
Yeah, so it turns out after genetic work that we did from teeth taken from these individuals, we can actually link them to the exact species, which is a species of bubonic plague, basically, a type of plague. And we don't know whether, so there are three different types of plague. There's bubonic, which is the famous, the buboes, the swelling under the arms and the groin. There's a pneumonic plague, which can go in your lungs, and which means I can actually give it to you by coughing. And then, of course, there's septicemic, which is within your bloodstream, which is the minute it hits your blood, it's 100% fatal. There's no chance.
Okay, so these two things are related.
Linked, yes.
They spread the same way?
We think they spread the same way. We think they began as rat, flea, human, right? So these fleas carrying the bacteria jumped onto black rats. Black rats made their way across through ships and through trade, through grain trade and so on and so forth. And as the rats started to die, because they also died from the plague, fleas that were desperate to get a meal started jumping onto humans and then spread them. And then as it moved into the lungs, then I didn't need the rats anymore. I could actually just give it to you or to the next person down the street by coughing. Okay. So we think, and that's why it spread so quickly.
Now every parent is like, reminder about why I swat flies away from my children's food.
Yeah, that's right. Many insects are vectors for pathogens.
Okay.
Yes.
The plague was indiscriminate in that it hit it. Didn't matter who you were, right?
It didn't matter.
Black, white,
no.
rich, poor, urban,
it killed
rural.
everybody. It killed everybody.
Right.
Yeah.
Okay. Do you learn anything from that it was just indiscriminate? Most diseases are, right, how they spread?
Well, most diseases, but in some cases it's not that the pathogen is discriminate, but the environment in which the humans live, it makes it a very different, right? So in some cases in developing nations and countries, for example, we know that the poor, unfortunately, live in towns where there's improper sanitation. And of course, for example, cholera then, right?
Yeah.
So, but otherwise, pathogens are indiscriminate.
Okay, because we always say that the plague hit everyone, didn't matter. And I'm always like, does that teach you anything as a scientist, that about that.
Very much so, yeah, absolutely. So knowing whether or not there's a preponderance in a particular environment or climate, or whether or not it hits water systems, and whether or not certain parts of the town have purified water and the other ones don't, that's an important epidemiological factor that then would be calculated into how do we estimate what the next pandemic might do or bring.
Okay, so you've got the plague.
I hope not.
Well, we're pretending here. You
Okay.
have the plague.
Yep.
It's in your lungs. You're
Yep.
coughing. Is that how I get it? You cough on me. It's
You could. Yeah,
I'm
you
sitting.
could
We're
get it.
sitting on the subway. I
Yeah,
get the plague from you.
Yeah. I cough.
as
You get it
easy as
into your.
that.
It can't, certainly if it's pneumonic. it can
Airborne.
be airborne.
In this case, it was not. It was bubonic.
Well, we think it was both bubonic and eventually became pneumonic. Well, That's
Okay.
why it spread so quickly.
So
through
that's how it
Europe.
spread. Is that just sitting next to having daily interactions with everyone? Why does it not? 40%. Don't get me wrong. Is obviously a huge percentage of the population.
I don't think anyone understands that. So, what do you think the percent of the population died from the Spanish flu, which is something in our collective memory?
There's hundreds of thousands.
Well, we had 50 million die.
50 million.
50 million from, but what was the percentage that it took out?
Percentage. 50 million. I don't know.
Like maybe 3%.
2%?
2%.
2%. Okay.
Yeah. So, and now we're talking about 40%. So, let's step outside the studio here, and almost every other individual drops to the ground. So you can imagine the bottleneck that humans went through in Europe and parts of the Middle East post this, right? This had a devastating effect.
Yeah, I mean,
on Europe.
I covered SARS 12 years ago.
Yeah.
If you think about that, it was probably like in our generation, the only time we've talked about this could be pandemic,
Right.
a human pandemic.
Yeah.
8,000 people
Yeah.
died around
Yeah.
the world.
As opposed to 40 million. Yeah.
40 million.
Yeah.
How come more people didn't die was going to be my question. I mean, what made this thing stop?
Well, that's $1 million question, which we're trying to investigate now. So, the easiest explanation is that some of us living in the population, because as a population you carry variation in your ability to ward off disease and infection, had some sort of protection against the plague, right? And so those that died were susceptible, and those that lived actually were protected against it. So a good example is, why is there a higher frequency of people living in Europe today that have resistance to HIV/AIDS?
You can tell me.
I will. So it turns out that we understood Nairobi sex workers have resistance to HIV/AIDS in Africa. And that involves a change on a receptor in a cell that's very interesting. It prevents HIV from infecting their cells. But the frequency of that gene, that deletion on the cell surface, turns out to be really high in Europe. And why? Because it looks like the entrance of plague or smallpox into the cells goes the same way via HIV does. So sometime in the past, Europeans dealt with plagues, and there was this massive culling off of the people that didn't have these changes. And those that did have these protective alleles, these genes that protected them, grew in frequency, because obviously those are the only ones left after this massive epidemic. Okay. So we really think it was that. It was some sort of protective immunity within humans that allowed certain individuals to sort of survive.
All right. And we talked about reconstructing genomes last night.
Yeah.
As well. Whatever it is, whether it's a virus or whether it's a large animal, what is the how do we do this? You have to explain it to me in layperson's terms, obviously, not in scientific terms. What is the methodology in reconstructing a genome?
Yeah, so it's fairly complicated, but it's fascinating that we can actually do it. So the first thing to do is to get a tooth from a victim and remove that tooth from a mandible. And why a tooth? Well, if we think that these infections were probably septicemic, so they were within the blood at some point, that means we know that within a tooth you have your root pulp, right? In your root cavity, and there's blood that's pushed up into the tooth. That's where the nerve endings are. No one likes going to the dentist, but so what we do is actually we drill up into that pulp cavity and we extract all the DNA that remains from the blood that's still stuck on the inside of those teeth. And then, not surprisingly, within that DNA, there's a pool that comes out. Not surprisingly, the DNA of the human is there because that was his tooth or her tooth. But the DNA of the microbes that naturally live within the mouth, we'd find in that blood. And then, of course, we'd hope to find the pathogen that killed the individuals. And so, not surprisingly, you find pestis, Yersinia, which is the bubonic plague bacteria, in that DNA. But again, like the woolly mammoth, it's all cut up into little tiny fragments. And so our job was, if that represents a minute fraction of that huge pool, how do we fish out the little tiny fragments that are just plague from all the rest of the junk that we're not really interested in right now and stitch it back together? And so what we do is we create almost fishing lines that are specific. So imagine if you went out to the ocean and you had a fishing line with a different color hook that was specific only to tuna. So you say, well, I don't want to hurt dolphin, I don't want to, and basically what we do is we create probes that capture only the bacteria of interest and leave everything else alone.
Okay.
So we can get rid of all the rest of the junk, and then we have only the bacteria of interest, and then we stick all the little pieces together, and we actually resynthesize the genome.
All right. So we had a photo up. That was actually an ancient tooth from a plague victim.
That's right.
Okay, so
Yeah.
that's the tooth part of this.
Yeah, that's right.
Let's bring up another photo.
Yeah.
This is a skull. I think we're bringing up. Yep, a skull
Yeah.
from the East Smithfield Plague Pits in London, which is where the Royal Mint actually.
That's right.
sits
That's
now.
where the Royal Mint
So
was
what's
there.
the relevance of this skull?
So this is one of the individuals that provided one part of a tooth that led to the complete genome of the Black Death. So this individual is a victim that was buried within this pit at East Smithfield. And the importance of this individual is we know the specific date when these individuals died. It was in 1348, probably January or February. And the reason we know that is because we have a decree from the king saying all the parish cemeteries are full because thousands of people were being buried every day. We need new land. And so he ordered this farmer's field to be purchased for the burial of these individuals, right? So that's one of the skulls that our collaborators at the Museum of London brought to us. We extracted the tooth, we extracted the DNA from that tooth, and we used that person's genes then to basically stitch this pathogen back together.
Wow. Okay. So we
Yeah.
got a skull, we got a tooth.
skull,
We're
we
going
got
to
a
bring up a
tooth.
couple more photos.
Yeah.
So here's this one. These are skeletal remains. This is from the East Smithfield site.
That's right. Yeah. So the East Smithfield site is again that plot of land that was purchased by the king for these massive mortality that was going on in London at the time. And it used to be the Mint. The Royal Mint was there. In fact, when the excavations took place in the 80s, you can still see all the copper that was actually flushed onto these remains. And they dug up. Fortunately, the Museum of London was allowed a relatively short amount of time to get out about 1,000 individuals, but there's still a lot of individuals still buried there. And today there's a big apartment complex sitting on top of those particular sites.
There you go.
So those individuals, we actually used many individuals to help us reconstruct multiple genomes at that point in time to see whether or not the diversity of that particular plague was different than the plagues that we see today.
All right. I'm mispronouncing skeletal. It's skeletal.
Oh, it
Telling
doesn't
me in
matter.
my ear. Okay. Let's bring out more skeletal remains then.
Okay.
Pictured. So these are partially buried. They're colored.
They're colored.
That's the obvious thing.
Well, yeah, so they're colored here to show that, so this is Justinian burial in Bavaria at a Roman, what was still an extent of the Holy Roman Empire at the time. And what's so interesting about this burial, so typically when we think of Black Death, we think of these mass burial pits. We think of hundreds of individuals, like in the East Smithfield, layer upon layer upon layer. And if you go back to the Justinian, about 800s before the Black Death, so 1,500 years ago, the way to look for pandemic sites is not these mass grave burial sites. You don't find those. But burials where you have more than one individual in a grave is usually an indication of everybody was dying at the same time, very rapidly. What was that, right? So here you have actually a family of those three individuals. You have the mother, father, and the red were actually the colors of obviously their child, right, that had died all in the same time, which is an indication of some sort of infection typically.
Right.
So that's why we use those individuals to help us reconstruct the Justinian plague.
And, okay, so the first three photos were Black Death. That last one is Plague of Justinian. What do you hope the bones will tell you about the Black Death? You just said what it might tell you about the Plague of Justinian, about how to... What do you hope those bones are going to tell you about the Black Death?
So I think there's two things that are really important in trying to understand from this research. One is we still have plague today, right? In fact, we know that plague is all over the world today, and that actually turns out to be because of the Black Death, right? Which is very interesting. I can explain in a second. But if we can compare the ancient plagues, both the Plague of Justinian and the Black Death genome, to the modern plagues, are there changes in genes that have affected its virulence or its subsequent attenuation? So it killed 40 million people. what were those changes that caused that? Was there something inherent in the bug that made it particularly nasty? Or was it just that us as humans, we were exceptionally susceptible? So here we have the best possible example. We have the humans that died of the plague, and then we have actually access to skeletons before the plague hit and after. And so we can address both the question of the genome of the plague, was it particularly nasty? And we can also use the requisite human skeletons to go, well, you know, were there changes in the human genome that allowed humans to survive the plague, and therefore it's actually lost its fierceness because we've evolved to actually counteract it by becoming more immune to it, so to speak.
Adapting.
Adapting, that's
Okay.
right. That's all part of evolution.
Now, you were going to make an earlier point, which everyone's a little bit hung up on, where you say there's plague everywhere.
There is.
There's a link.
There is a link, yeah. So interestingly, from analyzing the Black Death with the current plague, we see that they're all tightly related, and that the Black Death plague sits as the grandmother of all the plagues that we see across the globe today. So we had this massive epidemic of Black Death, and that actually reseeded areas of rodents that carry plague naturally and are completely immune to it with these new strains that came from the Black Death. You can't have, 40 million died. You probably had hundreds of millions that were infected. And so what happens is that gets back into the watersheds, it gets back into the bacteria, into the rodents that carry it naturally. So we think that Black Death spread back along the Silk Road to the highlands of China. And eventually it went down into Hong Kong and created this, what we know as the third plague pandemic, which doesn't have a fancy name like the Black Death. But on ships, basically, it went to Los Angeles in the late 1800s, early 1900s. It was called the Barbary Plague. So it was a plague. You could actually get $30 to kill rats in San Francisco at the time, because they figured those were the hosts. It also made its way to India, where it had a massive outbreak, and then from India down to Madagascar. And so whenever we hear about plague in a squirrel in BC, or if you remember several years ago, there was a plague squirrel in the park in Los Angeles that everybody was very scared
And they were
about.
worried about, in India there was a plague.
In India there was a plague,
Rats,
yeah. So all those have their roots in the Black Death, right? So all those are actually derived individuals from this ancestral plague that hit in 1345.
And yet, the panic of the word plague, obviously it has killed some people, not as many. So is that because of our adaptability? Is that what the thinking is, that we've just, like fighting off lots of different pathogens, that we've just become adapted?
I think it's multiple strains, multiple reasons actually. Something that we call syndemics. I think it has to do with the fact that typically most countries have proper sanitation, and so there's better health care, there's better access to medicines. I think partly it's because as humans we've adapted and have become immune to some aspects. I think the bug has lost part of its virulence because of a natural attenuation. If you think of any type of pathogens that's been successful, and it's always dangerous to call a pathogen successful in terms of human relationships, but it's evolved to not completely kill its host, right? So in Ebola, there's a very rapid mortality rate, but by doing so, it burns itself out. Mm-hmm. Whereas you think of something like HIV, which has had this long tenure in humans because it's attenuated its virulence a bit, and because we've repeatedly hit it with cocktails and drugs and so on and so forth, right?
I asked you this yesterday about de-extinction, and especially as it pertains to the woolly mammoth. Why do we want to understand? Why do we need to, I take your point that the Black Death sort of exists everywhere now.
Yeah.
But make the leap for me again from why we need to understand something so rooted in our past, it's an obvious question, to today.
Yeah.
What are we hoping to learn about us today?
Well, I mean, the Black Death and the Plague of Justinian, for example, are really important experiments in the past, right? We don't have access to pandemics today, and you certainly can't do a natural experiment like that to actually test what actually happened. So here we have these natural experiments that took place in the past. We have access to these skeletons, and we can start asking these very important and addressing these questions about why was it so deadly? What was it about the movement or the epidemiology of that particular pathogen that we need to understand so that we can better predict and be able to cope with pandemics that are inevitably going to come? Because it's always a matter of either a novel emergence or a re-emergence of something that already existed.
So it would help with pandemic preparedness, basically.
Absolutely, yeah. We want to be always at the forefront of understanding pandemics and infection and disease in general so that we are prepared for when it does strike, because inevitably they will. The question is whether or not we've actually spent the time and money. to be
And on this
prepared.
particular, as we try and figure out these links, are pharmaceutical companies involved? Because of course they would be involved if we had a pandemic. We saw with Tamiflu and all these things.
Sure, sure. Yeah, I think certainly as we move into preparedness and understanding what specific things in plagues or flus or Ebolas are critical for either for rapid screening or for drugs, I think there would be cooperation and collaboration between pharmaceuticals. Certainly at this point, we're just at the basic research stage. So
Right.
there is
And yesterday we also talked about, you mentioned about bringing back the flu virus.
That's right, yeah. Which is the only in a sort of strange way, the only de-extincted thing that we currently have, right? Which is this flu, this 1918 flu. And the reason for doing that was, for the exact reason, is that particular flu has been like no other flu we've seen since, right? Killing 50 million people, very high mortality by comparison to the date. So what was it specifically in that one? And so they've taken it one step further. They've actually reconstructed it, but then revived it and had, obviously in a highly contained laboratory. And if you look at it in rodents, when you give them that flu, they die within 24 hours. It's very, very frightening. So and they're starting to understand now why that is, right? So there's an actual benefit to
And would this
uncovering the
process
past.
be the same for bringing back the plague?
Well, yeah, there's a question as to whether or not we should. Yeah, in a contained environment, it might be very important to do, but you might be able to do it with genes rather than the whole entire organism, which would be safer in terms of, it's not something you want to let out, right?
Here's the million-dollar question, especially since SARS, I think in Canada, doctors and scientists have been, microbiologists, so saying there's going to be another pandemic.
Yeah.
I think we lay people are more attuned to this idea now that it will happen. It's just a matter of when. What is, I'm not going to ask you when, because I know you don't know,
I
right?
can't predict
It's a million-dollar question. But are we, in how we've adapted on many levels, but could a pandemic be as bad as the Black Death again? I mean, they were 800 years between the Justinian one and then the Black Death. And then we've lived for all this time without a pandemic with that kind of mortality rate.
Yeah.
When you look at the science behind what happened then and how we lived, I mean, are we looking at a similar situation, or is it every time we see a SARS or something, we're always like, the pandemic, Ebola was, was an epidemic, pandemic.
It did become a pandemic, and if you notice, one of the most important thing was sort of the delayed response was hugely problematic.
But I think people would say, look, we can just deal with this stuff now. It's not 1980, it's not.
Well, of course, we do have medicines. We do have relatively rapid response, of course, which we didn't, at the time you had plague doctors with large masks, right, coming onto the scene, and a lot more priests. In this case, we have medication, we have rapid response teams. In general, countries are willing to work together when there's a crisis like that. But nevertheless, it all depends on the pathogen, right? If we have something that's completely novel, and we have a scientific team that can move relatively quickly to sort of sequence its genome, trying to understand its biology, its pathology, and then rapid response in terms of production of drugs. But imagine, so if I was to send 100 people to the emergency room at any hospital in Toronto today, would they have a long wait?
Yes.
Okay, so imagine that we're talking about 10,000 people swamping a particular hospital because of a massive epidemic. Would the infrastructure be able to hold something like that?
No.
No. Okay, so.
But that's kind of odd. Like, we know that won't. We don't have the capacity.
No, that's right. So the question is, how do we build that capacity, or how do we build, structures in place to prevent the spread, right? So the best way to do is to be constantly prepared, like they do with flu seasons now. They have monitoring systems in places where we know that there's, pigs and chickens in various parts of Asia, for example. So we monitor for strains, and then we try desperately to close down trade routes and travel, which is almost impossible, but it's the right thing to do, right? It's the same reason why the CDC goes out and screens ground squirrels in the southwest of the United States for plague, because if those squirrels, those gophers, move into areas of human habitation, then there's the possibility of a jump onto humans, and you have a small plague outbreak in a city like Arizona, Phoenix, or something like that. So I think preparedness is the key, right? How do we screen, how do we stay on top, and how do we look for the possibility of new emerging diseases? But there's always some concern.
Thank you for 2 nights of a good conversation. Appreciate it very much.
Thank you very much, Peter, for having me.
Help TVO create a better world through the power of learning. Visit supporttvo.org and make a tax-deductible donation today.